Fertilization in flowering plants. Fertilization in flowering plants is the union of male and female reproductive cells to form a zygote, which develops into a seed. In flowering plants, fertilization involves the following key steps. 1. Pollination, the transfer of pollen from the anther of a flower to the stigma of the same or a different flower on the same or another plant. 2. Germination, once a compatible pollen grain lands on the stigma, the pollen tube germinates and grows down the style towards the ovary. 3. Fusion of gametes. 2 sperm cells are released from each pollen grain, one of which fuses with the egg cell within the ovule to form a zygote, while the other fuses with other cells to form endosperm, which nourishes the developing embryo. 4. Formation of seeds. The zygote develops into an embryo, while the ovule's other cells, including the endosperm, develop into the seed coat, providing protection and nutrition to the embryo. 5. Dispersal of seeds. The mature seeds are then dispersed from the parent plant, which allows the progeny to grow in new locations, avoiding competition for resources with the parent plant. Fertilization is crucial for the reproduction of flowering plants, ensuring the creation of new individuals that carry genetic variation, which is significant for ecosystem functioning and adaptation to changing environments. Gametogenesis in plants. Gametogenesis in plants involves the formation of haploid gametes, pollen grains and ovules, that unite during fertilization to form a zygote, which develops into a new plant. The process of gametogenesis in plants involves 1. Sporogenesis, the production of haploid spores through meiosis. In plants, this process occurs in specialized structures called sporangia, which produce either microspores pollen grains, or megaspores, ovules. 2. Gametophyte development, the development of the haploid spores into gametophytes, which are the structures that produce gametes. In angiosperms, the male gametophyte develops into pollen grains, while the female gametophyte develops into an ovule within the ovary. 3. Gamete formation, the production of haploid gametes within the gametophytes. In male gametogenesis, each pollen grain contains 2 haploid sperm cells. In female gametogenesis, the ovule contains 1 haploid egg cell and 2 haploid synergids, which function in pollen tube guidance. 4. Fertilization, the union of haploid gametes during pollination. In this process, the male gametes, contained within the pollen tube, reach the female gametes within the ovule, and fertilization occurs. This results in the formation of a zygote, which develops into a new plant. Gametogenesis in plants is crucial for sexual reproduction, enabling plants to produce a diverse range of genetically unique offspring, ensuring genetic variability within populations, and promoting adaptation to changing environmental conditions. Formation of male gametes in plants. The formation of male gametes in plants is called microgametogenesis or pollen development. This process is essential for the sexual reproduction of the plant, as it produces the male gametes, sperm cells, which are needed to fertilize the female gametes, egg cells. Microgametogenesis in plants involves several steps, including 1. Formation of the microsporangium. The microsporangium, also known as the pollen sac, develops within the anther of the flower and contains many diploid cells called sporogenous cells. 2. Meiosis. The sporogenous cells undergo meiosis to produce 4 haploid cells called microspores. 3. Mitosis. Each microspore undergoes mitosis to produce a vegetative cell and a generative cell. 4. Formation of the pollen grain. The generative cell and the vegetative cell form a structure called the pollen grain. 5. Germination of the pollen grain. The pollen grain is released from the anther and lands on the stigma of the flower. The pollen grain then germinates and grows a pollen tube, which carries the generative cell towards the ovary. 6. Fertilization. The generative cell inside the pollen tube divides and forms 2 sperm cells, which are released into the ovary and fertilize the female gametes. The process of microgametogenesis produces the male gametes necessary for sexual reproduction in plants, ensuring genetic diversity and variability within a population. This allows plants to adapt to their changing environments and ensures the survival of the species. Formation of female gametes in plants. The formation of female gametes in plants is called megagametogenesis, also known as embryo sac development or female gametophyte development. It involves the formation of the haploid female gametes, egg cells, that are necessary for sexual reproduction. Megagametogenesis in plants involves the following steps. 1. Formation of the megasporangium. The megasporangium, ovule, develops within the ovary of the flower and contains one or more megasporocyte cells. 2. Meiosis. The megasporocyte undergoes meiosis to produce 4 haploid cells, but only one of these cells survives and develops into the female gametophyte. 3. Formation of the female gametophyte. The surviving haploid cell undergoes mitosis to form a female gametophyte, also known as the embryo sac. The embryo sac consists of 7 cells, including the egg cell and 2 synergids that function in pollen tube guidance. 4. Pollination. The pollen grain lands on the stigma of the flower and germinates to produce a pollen tube, which grows through the style towards the ovary. 5. Double fertilization. The pollen tube releases 2 sperm cells into the embryo sac, where one sperm fertilizes the egg cell to form the zygote, and the other sperm fuses with the 2 polar nuclei to form the endosperm, which provides nutrients to the developing embryo. Megagametogenesis plays a crucial role in the sexual reproduction of plants, ensuring the genetic diversity and variability necessary for species survival and adaptation to changing environments. Distinct parts of the ovule during formation of female gametes are: during the formation of female gametes in plants, the ovule undergoes several developmental stages, and distinct parts can be observed. The ovule is the structure within the ovary that produces the female gametophyte, which contains the egg cells needed for sexual reproduction. The main parts of the ovule during megagametogenesis or female gamete formation are: 1. Megasporangium. This is the outermost layer of the ovule that surrounds the developing female gametophyte. It is also known as the nucellus. 2. Megaspore mother cell. This is a diploid cell located within the megasporangium that undergoes meiosis to produce 4 haploid cells called megaspores. 3. Embryo sac or female gametophyte. Out of the 4 haploid cells produced by meiosis in megaspore mother cell, only one will survive, enlarge and divide to form the embryo sac or female gametophyte. It contains a total of 7 cells, each with distinct roles. One of these cells is the egg cell, which fuses with a sperm cell to produce the zygote. 4. Integument. This is a tough outer layer that surrounds the megasporangium and the female gametophyte. The integument encloses the embryo sac and leaves an opening called the micropyle, through which the pollen tube enters during fertilization. 5. Funicle. It is the stalk that attaches the ovule to the placenta, tissue inside the ovary that supports the ovules. These distinct parts of the ovule are crucial for the proper development and maturation of the female gamete and play an essential role in sexual reproduction of the plant. Section of the anther and development of the pollen mother cell. The anther is the male reproductive organ of the flower in angiosperms, and it contains microsporangia that produce and release pollen grains. The microspore mother cell, also called pollen mother cell located inside the microsporangia, undergoes meiosis to produce haploid pollen grains. The following outlines the structure and development of the anther and pollen mother cell. 1. Structure of the anther. The anther consists of 4 distinct parts. The connective tissue. It connects the 2 lobes of the anther and provides support. The filament. It is a stalk-like structure that connects the anther to the flower. The theca. Anther wall. It is divided into 2 lobes, each chamber called a microsporangium or pollen sac. The microsporangia pollen sacs. They are the regions within the anther that produce and release pollen grains. 2. Development of the pollen mother cell. The immature anther contains diploid sporogenous cells, which divide by mitosis to produce haploid microspore mother cells or pollen mother cells PMC. Each PMC undergoes 2 rounds of meiosis, resulting in the formation of 4 haploid microspores. Each microspore develops into a pollen grain, which is a mature male gametophyte that contains 2 haploid sperm cells. In summary, the anther contains microsporangia, which produce pollen grains through the process of meiosis. The pollen mother cell PMC is the diploid cell within the microsporangium that undergoes meiosis to produce 4 haploid microspores, each of which develops into a pollen grain containing 2 sperm cells. The production of pollen is crucial in the sexual reproduction of angiosperms, as it leads to the transfer of male gametes to the female reproductive organ for fertilization to occur. Development of the embryo sac. The embryo sac, also known as the female gametophyte, is produced inside the ovule of angiosperms and contains the egg cell that fuses with a sperm cell during fertilization to form the zygote. The development of the embryo sac can be divided into 2 main processes. Megasporogenesis and megagametogenesis. 1. Megasporogenesis. In the ovary, a megasporocyte or megaspore mother cell located in the megasporangium undergoes meiosis, producing 4 haploid cells called megaspores. In most flowering plants, only one of the 4 megaspores survives, the others degenerate or are absorbed. The surviving megaspore undergoes 3 rounds of mitosis without cytokinesis, resulting in an 8-nucleate cell, which becomes the embryo sac. 2. Megagametogenesis. Within the 8-nucleate embryo sac, one cell migrates to the micropelar end and becomes the egg cell, while the other 2 cells located in the center of the embryo sac become the polar nuclei. The other 3 cells relocate at the opposite end of the embryo sac, forming the antipodal cells. The remaining 2 cells located on either side of the egg cell develop into the synergids, which play a role in guiding the pollen tube during fertilization. At maturity, the synergids and polar nuclei participate in the embryo sac's unique double fertilization feature, resulting in the formation of the diploid zygote and a triploid endosperm, which provides nutrition to the developing embryo. In conclusion, the embryo sac or female gametophyte develops from a single surviving megaspore that undergoes 3 rounds of mitosis to form 8 nuclei within a sac. This structure eventually gives rise to the egg cell, the polar nuclei, and other essential cells that participate in double fertilization for the sexual reproduction of angiosperms. Germination of pollen grains and development of pollen tube. The germination of pollen grains and development of the pollen tube are 2 essential processes in the male reproductive cycle of flowering plants that allow the transfer of male gametes to the female reproductive organs. 1. Germination of pollen grains. When pollen grains land on the stigma of a receptive flower, the pollen grain absorbs moisture and swells, rupturing the pollen wall and releasing 2 sperm cells from the pollen grain's cytoplasm. The 2 sperm cells are carried to the ovary by the growing pollen tube, where they fertilize the egg and central cell to form the zygote and endosperm. 2. Development of pollen tube. After the pollen grain lands on the stigma, a vegetative cell on its surface elongates into a slender tube, called the pollen tube, that grows through the style towards the ovule. During the growth of the pollen tube, it is facilitated by chemical signals between the pollen tube and the pistil, which attract and guide the tube. The vegetative cell provides energy and cellular materials to the growing tip of the pollen tube, while the sperm cells are contained within the tube's cytoplasm. The pollen tube grows through the micropyle of the ovule and delivers the sperm cells to the embryo sac, where fertilization takes place. In conclusion, the germination of pollen grains and development of pollen tube are crucial processes in the sexual reproduction of flowering plants. They allow for the transfer of male gametes to the female reproductive organs, resulting in the formation of the zygote and endosperm, which eventually give rise to the embryo and the seed. Fertilization. Fertilization is the process by which the sperm cell fuses with the egg cell, resulting in the formation of a zygote, the first cell of a new individual. In flowering plants, fertilization involves a unique process called double fertilization, which occurs when 2 sperm cells are released from the pollen tube and fertilize 2 different nuclei in the embryo sac. The process of fertilization in flowering plants can be summarized as follows. 1. Pollination. Pollen grains land on the receptive surface of the stigma and germinate, forming a pollen tube that grows down the style towards the ovary. 2. Double fertilization. The pollen tube penetrates the ovule through the micropyle and releases 2 sperm cells into the embryo sac. One sperm cell fuses with the egg cell, forming a diploid zygote, while the other sperm cell fuses with the 2 polar nuclei, forming a triploid endosperm nucleus. 3. Zygote formation. The zygote develops into an embryo, which will eventually grow into a mature plant. 4. Endosperm formation. The endosperm nucleus divides multiple times, producing a nutrient-rich tissue that provides nutrition to the developing embryo. 5. Seed formation. The zygote, endosperm, and other parts of the ovule develop into a seed, which contains an embryo and a food source enclosed within a protective seed coat. In conclusion, fertilization is an essential process in the sexual reproduction of flowering plants, leading to the formation of the zygote, endosperm, and eventually the seed. Double fertilization is a unique feature of angiosperms, which ensure the development of a well-nourished embryo and endosperm for the successful propagation of the plant species. Seed formation. Seed formation is the process by which the fertilized ovule matures and develops into a seed containing the embryo and a food reserve enclosed within a protective seed coat. The process of seed formation involves several stages that occur after the process of fertilization and includes the following. 1. Embryo development. After fertilization, the zygote divides repeatedly and forms an embryo. The embryo differentiates into various tissues, such as the embryonic axis, cotyledons and hypocotyl, root meristem, and shoot meristem. 2. Endosperm development. The endosperm nucleus of the fertilized ovule divides repeatedly, forming a nutrient-rich tissue that provides nutrition to the developing embryo. 3. Seed coat formation. The integuments of the ovule thicken and become hard and woody, forming the seed coat that encloses and protects the embryo and endosperm. 4. Maturation. The seed undergoes a period of maturation during which it accumulates nutrients and desiccates, becoming dormant and resistant to harsh environmental conditions. 5. Dispersal. Mature seeds are dispersed from the parental plant in various ways, such as wind, water, or animal dispersal, to new locations where they can germinate and grow into new plants. In conclusion, seed formation is a complex process that involves the fertilization of the ovule followed by the development of the embryo, endosperm, and seed coat. The mature seed contains all the necessary components for germination and gives rise to a new plant, ensuring the successful propagation of the species. Fruit formation. Fruit formation is the process by which the ovary of a flower develops into a mature fruit that protects and disperses the seeds formed from fertilization. The process of fruit formation includes the following stages. 1. Pollination. Pollen grains are transferred from the anther to the stigma, leading to fertilization of the ovules present in the ovary. 2. Ovary growth. After fertilization, the ovules present within the ovary develop into seeds, while the ovary itself grows in size, forming the fruit. 3. Seed development. The fertilized ovules within the ovary form seeds, which develop and mature within the developing fruit. 4. Fruit ripening. The fruit undergoes a series of changes that soften and enlarge the fruit, change its color, and develop its characteristic taste and aroma. 5. Seed dispersal. Once the fruits are mature and ripe, they are dispersed by various mechanisms such as wind, water, animals, or self-propulsion to ensure the successful germination of the seeds in new locations. In conclusion, fruit formation is the process by which the ovary of a flower develops into a mature fruit containing seeds, which aid in the dispersal and propagation of the plant species. Fruits play a crucial role in the ecosystem as they provide nourishment for humans and animals and play a vital role in seed dispersal and reproductive success for plants. The ovary of a flower develops into a fruit after fertilization. When the ovules within the ovary are fertilized by the pollen, they develop into seeds while the ovary wall grows and matures into a fruit that protects and disperses the seeds. The fruit can have a variety of shapes, sizes, and textures, depending on the type of plant species, and may be classified as simple fruits, aggregate fruits, or multiple fruits. Examples of fruits that develop from ovaries include apples, oranges, tomatoes, and strawberries, among others. Pericarp. Pericarp refers to the outer layer or layers of the fruit that develop from the ovary wall after fertilization. Essentially, it is the part of the fruit that surrounds and protects the seed, or pit, The pericarp, which may have one or more layers, typically consists of three main parts. 1. Exocarp or epicarp, the outermost layer of the pericarp that is often the skin or the rind of the fruit. 2. Mesocarp, the middle layer, which may be fleshy or fibrous, and forms the bulk of the fruit in many cases. 3. Endocarp, the innermost layer that directly surrounds the seed, and may be woody or stony in some fruits like peaches, plums, and cherries. The characteristics and functions of pericarp vary widely among different types of fruits, and its texture, color, aroma, and flavor are often used to classify fruits. For instance, some fruits like berries and drupes have fleshy pericarps that are consumed as food, while others like nuts and grains have dry pericarps that are discarded. The term pericarp is commonly used in botany and horticulture to describe the outer covering of fruits. Parthenocarpy. Parthenocarpy is a condition in which a fruit develops without fertilization of the ovules in the ovary. This means that the fruit can develop without the need for pollination and fertilization. In a normal fruit development process, pollination triggers the ovules in the ovary to develop into seeds, and the surrounding tissue develops into a fruit. However, in parthenocarpic fruit development, the ovary develops into a fruit even in the absence of pollination. Parthenocarpy can occur naturally in some plant species, while in others it can be induced by various biotic or abiotic factors such as hormone treatments, genetic modifications, or environmental stresses. Parthenocarpic fruits can be seedless or contain undeveloped seeds that are usually small and deformed due to the lack of normal fertilization. Examples of parthenocarpic fruits include bananas, cucumbers, seedless grapes, and some types of citrus fruits. Parthenocarpy is significant in agriculture because it enables the seedless production of fruits, which can have economic advantages as they are often preferred by consumers over the seeded varieties. Gibberellins. Gibberellins are a group of plant hormones that are involved in regulating various aspects of plant growth and development by promoting cell elongation and division. They were originally discovered in Japan in the 1930s, where a fungus was found to cause abnormal growth in rice plants, leading to their elongation and reduced yield. The active compound produced by the fungus was later identified as gibberellin. Gibberellins control multiple physiological processes in plants, including seed germination, stem elongation, leaf expansion, flowering, and fruit development. They work by promoting the breakdown of deleproteins, which are negative regulators of growth, resulting in the activation of genes responsible for cell expansion and division. In addition, gibberellins also interact with other plant hormones like auxins, cytokinins, and abscisic acid to coordinate plant growth and development. Gibberellins are widely used in agriculture as a plant growth regulator to increase the yield and quality of crops such as grapes, apples, and strawberries. They are also used in the production of seedless grapes, inducing the malting of barley in the beer industry, and promoting the growth of shoots in horticulture. Gibberellin biosynthesis inhibitors are also used to reduce the stem elongation of ornamental plants and turf grass. How to differentiate a seed from a fruit. Seeds and fruits are two different structures found in plants that serve different functions. A fruit forms from the ovary of a flower after fertilization and encloses one or more seeds. The primary function of a fruit is to protect the seeds and aid in their dispersal. Fruits can be fleshy, like apples and berries, or dry, like nuts and grains. A seed is an embryonic plant enclosed in a protective outer covering called a seed coat. Seeds are formed within the ovules of a flower after pollination and contain all the genetic material needed to form a new plant. The primary function of a seed is to generate a new plant and ensure the survival of the plant species. To differentiate between a seed and a fruit, you can examine their characteristics. A fruit typically has a fleshy or hardened outer covering that surrounds one or more seeds, while a seed consists of an embryo and a seed coat. Depending on the type of fruit, the outer covering may be edible or inedible. In contrast, the seed coat is always hard and typically inedible. Another way to differentiate a fruit from a seed is their function. Fruits protect and disperse seeds, while seeds are structures that have the potential to grow into a new plant. In other words, fruits are the protective structures that contain seeds, while seeds are reproductive structures that have the potential to produce a new organism. Revision exercise. 1. What is double fertilization? Double fertilization is a process that occurs in angiosperms, where two sperm cells fertilize two different cells in the female gametophyte. One sperm cell fertilizes the egg cell, forming a zygote, which develops into an embryo. The other sperm cell fertilizes two polar nuclei, resulting in the formation of a triploid nucleus, which develops into the endosperm. Double fertilization ensures that the developing embryo has access to the nutrients and resources it needs to grow and develop. 2. Explain what happens between the time the pollen grains land on the stigma and fusion of nuclei occurs. After pollen grains land on the stigma of a flower, they germinate and grow pollen tubes, which extend through the style and reach the ovules in the ovary. The growth of the pollen tube is guided by chemical signals from the female reproductive structures. Once the pollen tube reaches the ovule, it releases two haploid nuclei into the female gametophyte. One of these nuclei fuses with the egg cell, which leads to the formation of a diploid zygote that develops into the embryo. This process is known as syngamy or fertilization. The other haploid nucleus fuses with two polar nuclei in the embryo sac, forming a triploid nucleus that develops into the endosperm, a nutrient-rich tissue that supports the growth and development of the embryo. Between the time the pollen grains land on the stigma and fusion of nuclei occurs, The female reproductive structures undergo a number of changes, including the development of the ovary, ovules, and embryo sac. Hormones and other chemical signals play an essential role in organizing and coordinating these processes, ensuring that the timing and progression of events are synchronized for successful reproduction. 3. How are the following parts adapted to their functions? A. Stigma. B. Style. C. Ovule. A. The stigma is adapted to its function of receiving pollen. It is often sticky or has hairs that help to capture and hold the pollen grains. The surface of the stigma also has receptors that are able to recognize and respond to specific chemical signals, allowing for the selective acceptance of compatible pollen. B. The style is adapted to its function of providing a pathway for the growth of the pollen tube. It is a long, narrow structure that connects the stigma to the ovary, and is often lined with specialized cells that facilitate the transfer of nutrients and other materials from the style to the growing pollen tube. C. The ovule is adapted to its function of producing and protecting the female gametophyte, which contains the egg cell that is necessary for fertilization. It is often enclosed within the ovary, which provides protection from external threats like disease and herbivory. The ovule is also surrounded by protective coverings and the integuments that nourish and support the developing embryo, ensuring its survival until it can produce its own nutrients through photosynthesis.